Hello, and welcome to another lesson with Miss Washington. So, let's do a little recap. In Unit 1, we talked about the macromolecules, those large molecules that make up our body. And the four major macromolecules that we needed were what? Carbs, lipid, proteins, and nucleic acids. We talked about what atoms made up those macromolecules, right? So carbon, hydrogen, oxygen, and even nitrogen and phosphorus. We continued on and talked about in Unit 2, cells and the organelles or tiny organs inside those cells that help them to function. Just like your body has different organs in it, like the heart and the lungs and the intestines, your cells have tiny little organs in them, like ribosomes, mitochondria, and even nuclei, in order to help them function. In Unit 3, we talked about how these organelles actually carried out those functions to help us live. We talked about photosynthesis, cellular respiration, and even how our cells divided. And lastly, we just finished Unit 4, where we talked about our DNA, this wonderful nucleic acid that codes for our proteins, how it codes for everything we are and how our traits are actually expressed. Today, we're going to talk about how we inherit those genes. In Unit 4, we focused on the molecular structure of DNA, how it replicated, how it made proteins, how it coded for proteins. In this unit, we're going to talk about how this baby expresses those traits, how this baby or why this baby looks exactly the way that he does. So, welcome to Unit 5, Inheritance Genetics. In this first lesson, we're going to talk about how offspring can be predicted based off of inheritance patterns, based off of their parents' genotypes and phenotypes. So the essential questions you'll be able to answer after this lesson are, what are Gregor Mendel's laws of inheritance? And how are Punnett squares used to determine the probability of that offspring that will have a certain trait? So who is Gregor Mendel? That's the first question we need to ask. Well, Gregor Mendel was an Austrian monk. He studied science and math at university and worked in a monastery where he was in charge of the garden and taking care of plants. He began to question, why do some plants come out this way and other plants come out this way? Why do some plants come out shriveled while other plants come out smooth? Why do some plants come out yellow and other plants come out green? So he began conducting experiments, and he used pea plants to show how the traits are passed from one generation to the next. In his research, he came up with 3 laws: the law of dominance, the law of segregation, and the law of independent assortment. Each law helps us to explain how traits are passed from their parents to their offspring. So before we can begin, let's make sure we understand some basic vocabulary. Vocabulary is very important when we're explaining inheritance patterns. So what are alleles? Well, traits come in different versions, right? Alleles are different versions of a gene. For example, there is a trait for height. There is a trait for height. What are some versions of height? What are some different versions of height? Right, tall and short. So the trait is height, and the different versions or alternate versions are tall and short. So the allele for being tall might be a dominant allele, denoted by a capital T, whereas the allele for height, the allele for height that's short would be the recessive allele. And it's denoted or shown by a lowercase t. So this could be the allele, this could be the trait for fur color. That's the trait. And the alleles would be black fur, capital B, brown fur, lowercase b. Fur color is the trait. The allele, or the different versions of that color, would be black fur and brown fur. Dominant alleles are strong. They cover up recessive alleles. They're represented by uppercase letters. So as you see over to the right, the dominant allele for fur color in rabbits in this situation is capital B for black fur. Recessive alleles are covered up by dominant alleles. They're represented by lowercase letters. So the different version of fur color for these rabbits would be brown fur, and it's denoted by a lowercase letter because it's recessive. It gets covered up. So dominant alleles are always shown by capital letters, and recessive alleles are always shown by lowercase letters. So, recessive alleles are only expressed when there are no dominant alleles. So think about a dominant allele like a dominant personality in a relationship. In a relationship, this dominant person is always talking, always laughing, and always the center of attention. The capital letter always comes first when you're writing the alphabet. Whereas a recessive allele is kind of like a recessive person or a submissive person in a relationship. The submissive person doesn't really say much and usually follows the dominant person's lead. The only way that a recessive trait can be shown is when there are no dominant alleles present. So, now that we understand that alleles are different versions of a gene, let's understand that there are two alleles for each trait. Each offspring has two alleles for the trait. This is because they get one from their mom and one from their dad. So, if there was a bunny that had two capital B's, this will be considered homozygous dominant, a homozygous dominant genotype. Because it got a capital B from its mom, a capital B from its dad. Homo means same. So homozygous dominant means two same capital letters. This bunny would be black. Capital B, lowercase b, shows a heterozygous genotype because they have two different alleles. Hetero means different. However, this bunny would still be black. Why? Because it has a capital B, and dominant alleles always cover up recessive alleles. And the third kind of genotype is a homozygous recessive genotype. A homozygous recessive genotype is two lowercase letters, two lowercase alleles. So this bunny got a lowercase allele, a recessive allele from her mom, and a recessive allele from her dad. It is homozygous recessive because it is two same recessive alleles. Since this bunny has two recessive alleles, the recessive trait can thrive and flourish and show. So this bunny would be brown. So you heard me say genotype and phenotype at the beginning of this presentation, and you even heard me say it in the last slide. But what the heck is a genotype? Well, a genotype is an organism's genetic makeup, whereas a phenotype is the organism's physical traits, what we can actually see. The genotype plus the environmental factors that affect the organism equals its phenotype, so what it's going to look like. So the organism's genes and the environment that the organism is in determines how the organism is going to look. So the genotype of this bunny would be two capital B's. But the phenotype, the physical, what we can actually see, would be a black bunny. The genotype of this bunny is a heterozygous genotype because it has a capital B and a lowercase b. Its genetic makeup is capital B, lowercase b. However, its phenotype, its physical trait, what we can see, is still a black bunny because that capital letter overpowers the lowercase letter. So that dominant trait overpowers the recessive one and will always show. A homozygous recessive genotype, two lowercase b's, is the genotype, the genetic makeup. But the physical, the phenotype, what we can actually see, is a brown bunny. It's a brown bunny because there are no dominant traits. So both recessive alleles can finally show the recessive trait. So your genotype is your letters. Your phenotype is what you can see. So, how do we use Punnett squares to determine the possible genotypes of the offspring, the possible phenotypes and genotypes of the offspring from two parents? Well, we use a Punnett square to show or predict the types of offspring that will result from a cross of two parents. So how do you make a Punnett square? The first thing I would always like to point out is that we always need a legend or some type of key on our paper so that we know what each allele codes for and what each parent's genotype is. This can help us when we're later trying to understand the Punnett square. So how do you make a Punnett square? Well, the first thing you need to do is draw a square and divide it into 4 equal boxes. So you draw a square and divide it into 4 equal boxes. Then you have to place parent 1's alleles on the top. Parent 1's alleles or parent 1's genotype, look down here, are capital T, lowercase t. So we put capital T and lowercase t. That is parent 1's genotype. You put parent 2's alleles on the left side. Parent two's alleles are capital T and lowercase t as well. Now, you need to combine parent alleles in each box. How to do that? You bring the top parent's alleles down first. So the top parent, you bring the T all the way down, and the capital T all the way down, and you bring the lowercase t all the way down. Please note, I put my lowercase t all the way over to the right side in that box because capital letters always come before lowercase letters. Whenever you're writing the alphabet, you always write the capital letter first. You never write the lowercase letter. You never write it like this. You always write the capital letter first, and then the lowercase letter comes second. So because I had a recessive allele, this lowercase letter right here, I automatically put it over to the right because I knew it was going to be in second place. So whenever you're writing your alleles, whenever you're writing your genotypes, always, always, always put capital letters first. So you bring your first parent's alleles down. You bring your side parent alleles over. So I bring my capital T, and then I bring my lowercase t, lowercase t. And there, I have now completed my Punnett square. Remember, your uppercase letters, your capital letters must be written first. So we drew our square. We divided it into equal boxes. We placed parent one's alleles at the top and parent two's alleles at the left on the side. We brought down the first parent's alleles, the top parent's alleles, all the way down, and we brought the side parent alleles all the way over. Now, let's calculate the genotypic ratio. A ratio is just a comparison of stuffs to stuffs. So in this genotypic ratio, what are we doing? We're comparing the different genotypes to each other. So there are 3 different genotypes that we learned about: homozygous dominant, heterozygous, and homozygous recessive. There are 3 different genotypes. So now, based on our Punnett square, based on these 4 possible offspring, let's do our genotypic ratio. How many homozygous dominant genotypes do we have? 2 capital letters. How many homozygous dominant genotypes do we have? Right, 1. How many heterozygous genotypes do we have? Right, 2. And how many homozygous recessive genotypes do we have? 2 lowercase letters. Right, 1. So our genotypic ratio is 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. We're comparing the different types of genotypes to each other. The phenotypic ratio is determined by the genotypic ratio. So, two capital T's. What would be the physical representation of two capital T's? You know what capital T codes for, and if you know that dominant traits always show up, what would be the height of this plant? Would it be tall or short? Right, this is a tall plant. What about a heterozygous genotype? Right, it would still be a tall plant. Why? Because as long as there's one capital letter, that dominant trait will always overpower or cover up that recessive trait, that lowercase letter. So this plant would still be tall. And a homozygous recessive genotype, if you know that lowercase t codes for short, and there are two recessive alleles, so that recessive trait can finally show, what would be that phenotype? Right, short. So those are the three kinds of phenotypes, what we can see based on their genes. We can't see the genes in a bunny. We can't see our genes in our human bodies. We can only see what they physically show in the phenotype. And the phenotype is determined by the genes. So now that we know, in this case, two capital T's still code for a tall plant. A heterozygous or a homozygous dominant, two capital T's, codes for a tall plant. A capital T and a lowercase t, a heterozygous genotype, two different letters, still codes for a tall plant because that dominant trait will always overpower the recessive one. And two lowercase t's code for a short plant because now they can finally shine together. So, looking at these four offspring, physically, how many tall plants do we have? Right, 3. 1, 2, 3. We have 3 tall plants. And how many short plants do we have? Right, 1 short plant. Every time that we do a Punnett square from here on out, you will always have to label what each allele codes for. You will always have to tell me each parent's genotype. You will always have to tell me the possible genotypes and their phenotypes. You will always have to tell me the genotypic ratio and the phenotypic ratio. So there are 5 things, and you'll always have to show me your Punnett square as well. So you will always have to show me 6 things. So let's do an example together. You will see multiple examples like this throughout the rest of the year. So it says in dogs, the gene for fur color has 2 alleles. The dominant allele, capital F, codes for gray fur. And the recessive allele, lowercase f, codes for black fur. The female dog is heterozygous, and the male dog is homozygous recessive. So let's figure out everything that we need. So let's do our legend first. Let's do our key. So our key. The first thing you have to do is tell me what each allele codes for. So they gave us the dominant allele, codes for gray fur. So the dominant allele, capital F, codes for gray fur. And they told you the recessive allele, lowercase f, codes for black fur. So lowercase f, black fur. That's the first thing we had to do. The next thing is I want you guys to tell me the parents' genotypes. So I want you to show me the parent cross. It says the female dog is heterozygous. So if the female dog is heterozygous, it has two different alleles, then would her genotype be capital F, lowercase f, or lowercase f, lowercase f? Right. If she's heterozygous, she has two different alleles. So she would be capital F, lowercase f. So that's mom. And dad, it says the male dog is homozygous recessive. So if it's homozygous recessive, and we know that recessive alleles are denoted by lowercase letters, homo means same, what would be his genotype? Would it be capital F, lowercase f, or lowercase f, lowercase f? Right, he would be lowercase f, lowercase f. Homozygous, meaning two same, recessive, lowercase letters. Good. Now, let's do our possible genotypes. Our possible geno and pheno. So there are three possible genotypes and phenotypes. There's capital F, lowercase f, and lowercase f, lowercase f. Capital F are two dominant alleles. So what color would that fur be? Gray. Capital F and lowercase f, a heterozygous genotype, will still code for what color fur? Gray. Because there is that capital letter and that dominant allele always overpowers the recessive. But lowercase f, our homozygous recessive genotype, two lowercase letters can finally shine. So what color would that puppy be? Black. Good. Let's do our square. So, how to do our square? We put our first parent at the top and our second parent on the left side. Then you bring your top parent's alleles down. Notice I put the lowercase letter to the far right because they always come last. And then I bring my second appearance alleles over. And there we have it. We're almost done. Let's do our genotypic and phenotypic ratios. So our genotypic are our letters, what we can see. So do we have any homozygous dominants, two capital letters out of our offspring here? No. So it would be 0, capital F, capital F. How many heterozygous genotypes do we have? Right, 2. Because there are two different letters. Hetero means two different. So it would be 2, capital F, lowercase f. And how many homozygous recessive genotypes do we have? Right, 2. So 2, lowercase f, lowercase f. Now let's figure out what this physically means. We know what the genes are. Now let's see what that actually shows. So How many gray puppies do we have? Right, 2. So 2 gray. How many black puppies do we have? 2. This is how you're going to do a Punnett square every single time I give you one. You need to have your key, your parent cross, your possible phenotypes and genotypes, your Punnett square, your genotypic ratios, and your phenotypic ratios. You need 6 things every single time. All right. So the law of dominance states that a heterozygous organism with 2 different alleles will always express the dominant allele, because capital letters always, always, always cover lowercase letters for simple dominance. Number 2, the law of segregation says that during meiosis, the process of making those gametic cells, right? So during meiosis, an organism's 2 alleles separate into different sex cells. So you get a different combination every single time. So you never know what you're going to get. And the law of independent assortment says different traits on different genes are inherited separately. So they're independently assorted so that you get a different combination every single time. This increases genetic variation. This is called a dihybrid cross, where 2 traits are being crossed up. Look at all the different possibilities. Make sure to complete your comprehension questions. Thanks for watching.